Hello, I'm Jonas. First of all, I want to really thank the organizers for selecting my talk for presentation. It's really a pleasure to be talking to you, even though virtually. I'm a PhD student at the Quantitative Developmental Biology Lab at ETH Zurich, and today I want to talk about inferring regulomes from multimodal single-cell measurements with Pando. So in our lab, we're usually really interested in how cells change their identity over time. This is often a differentiation process, where a stem cell becomes differentiated into some mature cell type. Often this is depicted by these Waddington epigenetic landscapes, where the valleys control the state of the cell and form these bifurcation events while it differentiates. So inside the cell, this landscape is really shaped by a regulome, or a complex circuit of transcription factors that control a gene expression while the cell differentiates. At the level of a target gene, this regulation is controlled by regulatory elements, to which transcription factors can bind and then regulate collectively the expression of a target gene, which then gets translated, transcribed, and if itself is a transcription factor, it can also regulate the expression of other genes. And this really gives us this network character, where different transcription factors can regulate each other and form a circuit. So now with modern single-cell sequencing approaches, we can actually measure a lot of the interesting parameters of this process at the level of individual cells. And this gives us really powerful tools to infer these regulatory networks on a larger scale. So on the one hand side, we have single-cell RNA-seq. With this, we can obtain information about the transcription factor and target gene abundances by counting their transcripts in each individual cell. On the other side, we can also obtain information about the binding sites themselves by using single-cell attack-seq, which measures the chromatin accessibility across the genome. And now, since recently, we can also actually obtain both of these measurements from each individual cell. However, most gene regulatory network inference methods that we were aware of really only take the RNA-seq modality into account in their model. So they would build some statistical or machine learning model to describe the expression of a target gene based on a function of the transcription factor abundances. However, one can see that actually the expression of a target gene is not only dependent on the transcription factor abundances themselves, but on their interaction with the binding site. Because even if a transcription factor is highly abundant, if the binding site is inaccessible, it can also not bind and not regulate the transcription of a gene. So we thought that it would be great to extend this framework to really also take into account the accessibility of the binding site directly into the model, and thereby really make use of these multi-omic measurements where we can obtain both modalities from each individual cell. So this was our motivation to develop Pando, and it works as follows. First, we consider the peak regions around the regulatory region of a target gene. We can further narrow down these peak regions by considering conserved elements and candidate cis-regulatory elements from ENCODE. By taking the union of these two elements and intersecting them with our peak regions, we obtain a set of candidate regions. In these candidate regions, we can now scan for transcription factor motifs, and this is really to pair up each transcription factor with its potential binding site. Now, since this is kind of multi-ohm data, and we actually have also the expression of all genes and the accessibility of all peaks for each cell, we now have everything together to fit our model. Here we use a linear model that models the expression of the target gene based on the interactions of the expression of the transcription factors with the accessibility of their binding site. Fitting this model gives us these parameters beta, which are really a measure of how these transcription factor binding site pairs regulate the expression of a target gene. Fitting these models for many genes gives us essentially a network where each node is a gene and each edge is characterized by one of these coefficients. So this pipeline I just showed you was actually implemented as an R package. It's available on GitHub. It's still quite in its early stages of development. We're really actively developing it and working on it. But we really tried our best to make it easy to use and make it really work smoothly with a lot of the most widely used single-cell analysis tools in R, namely Sera and Signac. So now I want to go into a little bit of a story on how we applied Pando to our own research. So here we applied Pando to a multi-omic atlas of cerebral organoid development. When we grow cerebral organoids, we start from stem cells, we grow them in 3D, and then we culture them in a medium that makes them become neurons. And that really gives us these brain organoids, these heterogeneous neuronal tissues that can form different neuronal types. On the right-hand side, you can see a UMAP representation of this, where we start from stem cells on the left, and they then differentiate out into these diverse neuronal populations on the right. So for this data set, we actually didn't measure multi-omic measurements from each individual cell, but actually the cell suspension was split into two, one for RNA-seq and one for ATAC-seq, and we then integrated them to obtain pseudocells with those modalities. So that's for sure also possible. In this dataset, we can now identify these trajectories that lead up to these brain region-specific neurons. So first from stem cells, cells go down this differentiation path and first bifurcate into non-telencephalon and telencephalon. Telencephalic progenitors then further bifurcate into dorsal telencephalon and ventral telencephalon, finally leading up to these brain region-specific neuronal populations. We were really curious how this diversity is set up and what are the regulatory mechanisms behind this diversification. So we used Pando on this dataset to infer a regulatory network in these cerebral organoids. This is one representation of this, where each transcription factor is a dot, and we can actually further summarize this network to the level of transcription factors. One classic thing that one would do is to find these so-called gene or transcription factor modules, where a transcription factor is actually represented by the set of genes that it's inferred to regulate. However, since we are incorporating also the binding sites directly into the model, we can also summarize these what we call regulatory modules, where a transcription factor is essentially characterized by the set of binding sites that it can bind. And taking these two modules together really gives us a good view of the regulatory and the genetic programs that change during bifurcation into different lineages. On the right-hand side, you can see the differences in module activity between the dorsal and the ventral telencephalon, and we can actually see that the branch specificity is correlated between the gene modules and the regulatory modules. Especially for the dorsal telencephalon, we can now also see some transcription factors whose gene modules and regulatory modules are really highly specific to the dorsal telencephalon. Now, because we're including these binding sites into the model, we can also really zoom in and look at these inferred regulatory regions specifically. So here, we're looking at an upstream region of a transcription factor, EMX1, which is specific to a dorsal telencephalon. We can actually see a lot of these inferred regulatory regions and the transcription factors that bind them. And one thing that I hope you can appreciate, that a lot of these regulatory regions also really drastically increase in accessibility once we come into the dorsal telencephalon. Now lastly, I want to tell you about how we use this GRN to interpret the results from a genetic perturbation experiment. Here, we knocked out GLI3, a transcription factor that's involved in dorsal telencephalon development. We knocked it out in stem cells and grew organoids from these knockout cell lines. We performed single-cell RNA-seq, and then we did what one would usually do, and that is perform differential expression analysis between the knockout and the wild type. What you would then get is this list of genes, and you don't really know how these genes are related to GLI3. So we thought that it would be great to really build this mechanistic model with the help of our GRN of how GLI3 regulates these genes that we see to be differentially expressed. And on the right-hand side, you can actually see this is a subnetwork of our GRN, where each edge is fully consistent with what we observe to be differentially expressed. One thing we can actually see here is that a lot of these genes are not directly regulated by GLI3, but actually secondary effects that are affected by some other gene that GLI3 down or up regulates. We can also use the results from this knockout experiment to validate our gene regulatory network. Here we can see that the DE genes also globally are actually highly enriched among the direct and the indirect targets of GLI3. And that also the direction of the DE, at least for the direct targets and the most highly differentially expressed genes, is actually fairly well captured by our gene regulatory network. With that, I'm at the end of my presentation, and I want to thank everyone who was involved in the project. First and foremost, Sophie, who really spearheaded all of the experiments and produced all of the cool data that I showed you today. Chi-Song, who really brought in some great ideas and really helped out a lot in discussions. Xianhui and Tamash were the beta testers of Pando, and they're also now using Pando in their own research. Barbara and Gray, of course, for their excellent supervision. And last but not least, the Boehringer Ingelheim Funds for funding my PhD. Thank you.